Okay, thanks everyone for joining us today. My name is Surabhi. I am a postdoc with Lena Teng at Emory, and I would like to introduce our speaker today. We have Professor Wilson Joyner, who is in the College of Biological Sciences at University of California, Davis. Dr. Joyner obtained his PhD in biomedical engineering from Johns Hopkins University, where he studied error feedback in predictive eye movement. From there, he went on to do a postdoc at Harvard University and the National Eye Institute. He received the NIH K99 Pathway to Independence Award, and he has also since received the NSF Career Award for the Biomedical Engineering Program. His lab now at UC Davis is interested in studying how external and internally generated sensory information is integrated in healthy individuals and in comparison to certain disease and impaired populations. Specifically, he studies people with schizophrenia, Alzheimer's disease, spinal cord injury, and upper extremity amputees. His talk today will be about the latter population and is titled Examining Dexterous Motor Control in Children Born with Below Elbow Deficiency. Go.
Thank you for that kind introduction. Good afternoon. I want to thank Lena for giving me the opportunity to talk to you all today. I also want to apologize that if I yawn, it's only because of the time difference and daylight savings time. My body is rebelling, so I apologize. But right, the title of my talk today is examining dexterous motor control in children born with a below elbow deficiency. This work is in collaboration with Shriners Hospital for Children and also some colleagues at George Mason University and some colleagues in the Mechanical and Aerospace Engineering Department at UC Davis. So I wanted to start with the problem first that we're kind of attacking. So when it comes to upper limb prosthetics, there's been a lot of improvement, a lot of work that's been done to make really advanced, really capable hands, really capable upper extremity devices. But the control of those devices is still in its early stages. At least there's still some improvements that can be made. I shouldn't say early stages. Still some improvements that can be made. And here's an example. So this is a patient who recently had a traumatic event and there was an amputation, and he is having a conversation with a prosthetist on basically how to learn how to control this robotic hand. And I want to kind of have you focus on what he says and the conversation they're having back and forth. I'm hopeful that the video is going to be loud enough. So, and that's where it stops, or at least that's when the video stops. And I wish that it kept going because the process is about to say, like, the human hand, I want him to say, like, the human hand doesn't work this way. But part of the confusion that the patient is having is like, so I stop activating my muscles and then the hand will stop at, this position. And there's definitely some amount of confusion in that question he's asking the prosthetist. And kind of diagram out the issue with this is, some of these prosthetics, some of these upper limb prosthetics basically work where if you are relaxed, if you're in some relaxed state, it's recording, let's say the electrical activity of the upper limb, muscles and the residuum, the prosthesis could be in a, let's say, closed state. So the hand could be closed. To open it up, you have to activate the muscles. So you have to activate, let's say, the extension muscles. You activate your extensors of your forearm, and the hand will open up, and it'll keep opening typically until you basically stop, until you basically go back to a relaxed state. So when you go back to a relaxed state, now the hand, prosthetic hand, is in a different state, but you have the same muscle states, right? So relax here and relax here. There's two different states for your prosthesis. Am I gonna go? No, maybe not. Oh, it is gonna go. It's gone. It's totally gone. All right. It still works. All right. To have the hand close, you do the reverse muscle activation. So if you activate the flexors, you can now close it. But this idea of having, two different states of your device, closed or open for the same state of the muscles is not intuitive to learn. It's not something that our hand naturally kind of does when we operate that. To give another example, probably more effective example. Here is a video I borrowed from the laboratory for bionic integration at the Cleveland Clinic. And this patient has a very advanced prosthesis, so it can do all sorts of articulations, all sorts of maneuvers. And the job of this participant is fairly simple. It's just to pick up these pins and place them on this rod. And you can see that she's having quite a bit of difficulty getting the hand in the right position, basically reaching and grasping these pins and then putting it onto this metal rod. And the hand is actually going to do some very non-physiological things. Like I think in a couple seconds, it's actually going to, right. So we don't really put pins on beams like that. And of course the hand's going to do, I think, a complete 360 right here, which of course our hands can't do. So learning how to do that, learning how to control these kind of devices is really difficult. The device themselves are very, very advanced, but the control signals, or at least the way that we can control them, is fairly complicated. But it doesn't always have to be this way. So this is Isabella. This is her as an infant. She was born with a congenital limb difference. So she was missing part of her left limb. When I met her, she was a 10-year-old in Northern Virginia. So this was at my prior institution at George Mason University. And she and her mom came to the university and approached the engineering school because they wanted to know if we could build a fairly simple prosthetic, 3D printed device to allow her to play the violin. And I had a senior design team at my prior institution that worked with her, worked with the School of Music, looked over the mechanics of playing the violin. They spent a tremendous amount of time with her in formulating this device to fit her limb. And her favorite color was hot pink. And they built this prosthetic for her, and totally passive. So there's no electronics in this. And this is her using the prosthetic to play the violin. And I would argue that that's pretty good, right? So again, fairly simple device, but there's a lot of very sophisticated motions and movement that she can basically use to control that fairly simple prosthetic to play a fairly sophisticated instrument. And I just wanted to put up that the Washington Post thought so too, and they wrote a nice story about her and the senior design group that basically worked with her. So prosthetics don't have to be non-intuitive. They can be very intuitive. They can allow you to do obviously very sophisticated things. And one of the things that we're trying to do is like, well, how can we have better control signals? How can we have better ways of basically controlling these devices? And the way that we've been going about doing it for the past 10 years or so now is using ultrasound imaging. So most of us are probably familiar with ultrasound imaging. If you have an ultrasound transducer, you can look at changes in the muscle structure in the body. If you've ever had a scan of your chest, of your heart, if you've ever had kids, you've seen plenty of ultrasound. But ultrasound imaging can visualize the main muscles and their functional compartments in the forearm too. So if you have an ultrasound transducer on the forearm, you can see the various muscles of the forearm. You can look at various bones, the ulnar and the radius of the muscles of the limb, and you can basically see the different changes as folks try to basically activate those muscles. So sonomyography, bless you, is the real-time change of muscle architectural parameters, the real-time changes of the structure of the muscles that you can obtain using ultrasound imaging. And we can basically apply ultrasound imaging to folks that have some kind of limb difference, either traumatic or some congenital limb difference, basically get an idea of like what kind of, what is their motor intent? What are they trying to do by activating their muscles? To give you a sense of basically how we can apply this, here's a fairly simple example. So the photo on, let's say on the left, right, is index finger flexion, and the one, no, sorry, on the left is middle finger flexion. The one on the right is index finger flexion, and we're just asking an able-bodied subject to basically do a very simple motion. So flex their index finger or flex their middle finger. And you can see all the different components of the forearm. So you can see the muscle components. You can see the nerve, the median nerve there. You can see blood vessels, things like that. And if you take the ultrasound imaging well, folks are basically trying to do this fairly simple, flexion of these two fingers, even though the fingers motions are fairly similar. You're talking about index finger flexion, middle finger flexion. The changes in the images are actually very different. I'm going to play the video. If you look at the median nerve, the one that's marked there, if you look at the median nerve, you can see the median nerve is doing something very different in both those images for fairly simple, fairly similar motions of these two fingers. So the median nerve, for index finger flexion is kind of going down, maybe slightly to the right, but in the middle finger flexion, it's definitely going down even more. So there's definitely different changes that are taking place in these images for, again, fairly simple movements of the fingers. And we can look at specific spatial temporal patterns of these muscle deformations and basically associate them with different movements. So we can image the muscles of the forearm and basically decode that into what the motor intent of these subjects are. To give you a breakdown, basically, of how we apply that to our patient population, kind of this kind of like time theory here. So you have the muscle activity, right? You can do some dynamic imaging using ultrasound imaging. We can do some basically some processing of those images, and we can look at like different places of where the images are changing the most, where they're changing the least. We can decode that into different types of motor intent, and then hopefully we can use that for control of these devices. So the way that we apply this, again, to our participants is ultrasound imaging is sampled at a fairly low sample rate, so 30 frames per second. Transducer frequency is 11 hertz. This is one of my graduate students. You can see the ultrasound transducer right here on his forearm. And in this case, he's just doing a fairly simple power grasp. So he's going from a relaxed state to basically doing a power grasp of his hand. And we can look at the images of the ultrasound, and we can look at differences. Where are the images changing the most? Where are they changing the least? So we take those images, we shrink them down to basically kind of compress the information, shrink and filter each frame. The raw data is roughly 1,000 pixels by 1,000 pixels. We reduce that down to about 100 by 100 pixels, 128 by 128 pixels. And then we can do some fairly simple analysis of those images to essentially create a QR code. So if we have consecutive images, right, from the ultrasound transducer, we can look at where are those differences taking place? Where are we seeing the most changes from a relaxed state, let's say, to a power grasp? We can threshold where those changes are taking place and basically make everything where, above that threshold, let's say, is white, below that threshold is black, and we essentially have a QR code that represents power grasp. So this is the representation in this two-dimensional space, the changes that are taking place in the ultrasound imaging. This represents a power grasp. And of course, we don't just end at power grasp. We can look at all different types of, all different types of motor intent. And we can use machine learning to then go through, is like, all right, for this particular image coming in, or these, these changes of the ultrasound images coming in, what is it best match? What is it best basically, similar to in terms of our, let's say, our library of different QR codes for different types of motor intent. So we can use a fairly simple algorithm, k-nearest neighbor algorithms to basically figure out, like, if this unknown image is coming in our library of different motor representations, what is it best matched to? What is it most similar to? We can continually adjust this performance. So we can continually try to figure out, all right, improve basically our decoding of like what these images coming in basically are most similar to in our library. And we can test each muscle state, we can treat each muscle state, each grasp pattern like a unique QR code. So we can basically go through and match these incoming images to our library of QR codes for different types of hand grasp patterns. So far, does all that make sense? Question?
Them from each other. So like other like stereotypical like movements or something, like I say, okay, right now I have a complete basis of all the possible movements I can have.
That's an excellent question. In a little bit, I'm going to get into confusion matrices and basically how well we can classify these different types of codes, but I think kind of gets at your question a little bit. But that's also some really good computational questions too, or at least analysis questions, because you want to know how separable these things are. So like a supplementary slide, but that's a great, excellent question. All right. Anyone else? Yes.
like person-to-person variance in like achieving these sort of like muscle states?
Right. So that's an excellent question too. When we get into the kids, all the kids have very different structures in their residuum. The traumatic also, their structures are different from person to person. So these are somewhat more personalized and more generalizable kind of approach. But that's a good question. Like that is something, again, another good analysis question of just like, how similar are these representations from person to person? Yeah. Okay. Yes. Yep.
I mean, something to come from what's like change and what
Yes.
Yes, so yeah,
I think I missed the first part. Can you say the first part again? Can you say the first part of your question again?
was thinking about the muscle length change.
Yes.
There is something coming versus something missing.
Yes.
I don't know where muscle offense is like anything. I understand that I don't know how muscle
That is true too. So there are various aspects about the dynamics of the change that would also be entirely, I shouldn't say entirely separate questions. There'd be a very relevant question. But in terms of what we're trying to do is trying to understand motor intent. But if we're talking about the dynamics of the muscles, or at least the characteristics of those muscle changes, that would be a very relevant, but also somewhat of a different question than what we're trying to get at in terms of like their control over those muscles. Excellent question. It's just not something that I've convinced a graduate student to study yet. But still, excellent question. Okay. All right, to give you an example of how this works in practice, this was at George Mason University. This is a adult with a traumatic limb loss. You can see the ultrasound transducer on their residuum. We are recording the imaging of those residual muscles, and we're decoding that information, and then we're showing her a simulated hand to basically tell her, this is what we decoded based on your intent. You're going to hear her say, this is what I'm basically trying to do with this missing hand, and we're going to show her, how accurate we are. So that hopefully made sense, right? Okay. So in the early studies, the ones we did in adults, we basically concentrated on three groups. So the first was able-bodied. So we did this study in able-bodied subjects. This is a confusion matrix. On the y-axis is the predicted movement. So what we're basically decoding, what we think the subject is trying to do. On the x-axis is the actual intended motion. And it just gives you a sense of how well, out of the number of times that we basically ask the subject to basically, enact a motion, how well were we able to correctly classify that motion correctly. And you can see for the able-bodied subjects, we did a fairly good job. There's only a misclassification for key grasp, about 4% for power grasp. So the able-bodied subjects were able to do that quite well. It's definitely more interesting when you get to the patients with a limb difference. So subjects with a traumatic amputation, I think there were 5 patients that we tested. Again, intended motions on the x-axis, the decoded motions are on the y-axis, and you can see that we do a fairly good job for these 5 different hand grasp patterns, decoding the motor intent of these subjects using ultrasound. Roughly, most of these are above 95% in terms of the accuracy of decoding. You can definitely see that there are some misclassifications, right? So again, key grasp, point are misclassified across the board, really, to certain other hand motor intent. But we are overall doing a fairly good job of decoding the motor intent of these traumatic patients using ultrasound. A more, at least in my mind, a very interesting aspect of that, first study was this one subject, this one patient that had a congenital limb difference. So this is an adult who was born without a hand, and we are decoding the motor intent in this adult subject for hand grasp patterns in a hand that never existed, right? So again, on the y-axis, we have all these different, decoding of motor intent. On the x-axis is what they were actually trying to do. And we still do a fairly good job of decoding these different types of motor intent in someone who didn't have a hand, right? So this is an adult. Bless you. The next question, or at least the question we asked, yes.
So in all of these cases, could the subjects see the screen?
The ultrasound screen.
Even like, the video that you just showed.
In these cases, no. That was just a demonstration of like how well we were actually able to code. In these,
So they're able to do, like, these
Yeah.
are all without visual.
all without visual feedback. But we're getting to visual feedback a little bit. That's a good question. Yes. Oh, that's a good question too. One hand. But the question is, if it was both hands, what would happen? That's a good question. Yep. So the thing that we asked next was, if you were born without a hand, what does that look like? What does the control look like? Do you actually have control over these residual muscles that you actually know what different hand grasp patterns are? So we asked that question in kids. When it comes to kids, they are very different than your adult population when it comes to limb differences. So unlike adults, acquired amputation is much less common in kids. If you have a limb difference as a child, it's probably something that you were born with, as opposed to something that unfortunately you had like some kind of traumatic event or some traumatic accident that caused that limb difference. The most common condition is a unilateral congenital below elbow deficiency. We call it UCBED for short. So there's a limb difference that was congenital since birth. It may appear similar to amputation. So if you look at these two pictures, it definitely appears similar to an amputation. But these children were born with this limb difference. They're also a very unique population because there's a limb difference, but the other unaffected side develops and is controlled normal. There's no difference in the there's no difference from a able-bodied subject when you talk about the unaffected side. That affected side though has this limb difference. Current commercial devices, and I think there are a number of people in this room that would probably agree with this. Current commercial devices don't provide sufficient functionality, and pediatric devices abandonment rates are very high. When you look at the kids, their abandonment rate is 35 to 45%. That is probably about 10 to 15, maybe even 20% greater than what you see for adults. One of the reasons is these are kids that were born with that limb difference, and they've gone their entire life basically compensating for that. They have found a way basically to operate around the world with one hand. When we talk to the surgeons, our clinical collaborators, the surgeons basically say like, yeah, they don't really have a sense of loss because they have learned how to operate with one hand. And in fact, when you think about it, a lot of the things that we do, in day to day, we definitely have bimanual, coordination, bimanual movements, things like that. It's obviously very important, but you could probably get by with one hand for most of the things that we do every day. And they have found some compensatory ways of doing that. But the abandonment rates are really high. And one of the reasons the abandonment rates are really high is because the devices that we have offered them are not particularly effective. We need to understand the ability to control these muscles, the ability to actually control the residual muscles of their limb in order to actually provide devices that actually meet their needs, that are actually an improvement over, just going about using one hand to interact with the world. And understanding this patient population is a challenge. There is definitely a number of things that are difficult in studying these kids and this particular condition that makes it difficult to basically understand, or at least wrap our heads around what their abilities are. In terms of the difficulties of studying kids, I can point to my own kids. I have 3 kids. Children grow, surprisingly. They get bigger. Their muscles develop with age. They start having opinions about things. And these are my 2 oldest kids. So on the couch is Marie. The middle child is Neva, who's standing. And if you just take a look at her, the oldest trying to give a Cheerio to the middle one, you could see like her coordination, her ability to basically reach out and grab that Cheerio is all over the place. She is not very good, right? But like I said, children get better, right? They get older, their limbs get longer, they get better coordination, integrating of sensory information. This is them 10 years later. Marie still on the left side, Neva on the middle, and obviously they're much better, right? All right, and we had a third kid. So the third one actually wanted to get into this as well. So right, so you definitely have improvements over age, over development. They definitely can get better at this. When I showed my wife this, was going to be part of my presentation, one of the things that she pointed out was like, this is the one on the right side. That's on the East Coast. This one is on the West Coast. These are obviously 10 years apart. The rug is the same. So she has pointed out this is even more evidence that we need better furniture. Okay, so to what extent does the missing afferent input, the missing hand, influence motor control? So that's one of the things that we're trying to answer. You know, if you don't have a hand, how well can you basically control, you know, these muscles and basically enacting different types of motor patterns? How does this affect their ability to control a prosthetic device? So that is ultimately the thing that we're trying to answer. Like if you don't have a hand, if you actually haven't actuated a hand, how do you know what power grasp is between a point, between a pinch, things like that? So to do this, we have been partnering with Shriners Hospital. It's been a very successful collaboration. It's been imperative to the things that we've been trying to do. Much like Isabella, we've been studying a number of kids that have a unilateral congenital below elbow deficiency. There are a number of kids that are treated by Shriners Hospital that we have been very fortunate enough to have the study and basically examine. And the objective, again, is to understand the capabilities of children's affected muscles. We were looking at kids between the ages of 5 to 20 years old. Shriners Hospital does qualify 20-year-olds as still children. So we're looking at a broad range of ages. We're also looking at different sites. So we have the main site in Sacramento, just down the road from UC Davis, but we also go to Portland, Chicago, and Greenville to basically study a number of kids. The goal, this is a 3-year study. We're 2 years and a couple months into that study. The goal is to have data on 50 kids by the time we are done. To give you an example of the results that we have so far, this is one patient or one subject. This is a 12-year-old. On the y-axis of this confusion matrix is the actual motor intent of this 12-year-old. On the x-axis is the predicted, so the decoded motor intent, again, using ultrasound. This is in the, again, the limb that was affected, so it doesn't have a hand. And you can see that we are fairly good, again, at classifying these, in this case, 10 different hand motions or hand motor intents of this individual. So this is someone who's 12. Here are the results comparing both the limbs. So we call these spider plots. So basically, along the circle are the different types of motor intents. So you see pinch, key, things like that. The radius of that circle gives you an idea of how well we can basically decode the motor intent. So if it's at the center, that would be 0. So it would be really bad, obviously, in decoding. If it's all the way at the end, if it's at the edge of the circle, that would be 100%. So we'd be able, do it near perfect. You can see that the red is the unaffected limb. So that is, again, the limb that develops normally. The blue is the affected limb, and you can see that they overlap quite well. So we're able to decode the motor intent of these 10 different types of hand grasp motions, and the affected and the unaffected limb fairly similar in this individual, this 12-year-old. If we look at someone who is slightly older, so we go from 12 to 16. Again, the confusion matrix on the top, the spider plot on the bottom, and you can see again there's quite a bit of overlap between the affected and unaffected limb. We don't see much of a difference in our ability to decode the motor intent between the two limbs. Again, using ultrasound. If we go younger, so that's 12, that's 16. If we go younger to 8, again, you see a fairly significant overlap between the affected and unaffected limb. So we're able to decode the motor intent in these individuals across ages fairly well using ultrasound. We don't see much of a difference. One of the questions I typically had is like, you've been showing us all this information about ultrasound. What about EMG, the normal way that you actually would decode these things? We've done the EMG studies as well. So we've put the EMG sensors on these kids and looked at our ability to decode motor intent using EMG. This is example for one individual. This is the unaffected limb. And you can see in the confusion matrix, they're able to code the motor intent fairly well. And the unaffected limb in this individual, we see a little bit of a drop off in the affected limb, not as good as what we saw for ultrasound, but again, in a limb that never actuated a hand, we were able to decode the motor intent using EMG signals quite well. So ultrasound, EMG, different sensing modalities were able to decode motor intent in a somewhat similar way. If we look at age, that's also really interesting. So here's classification accuracy by age. Again, the same kind of spider plot, the same kind of structure of that plot. 0 would be not being able to decode motor intent well at all. If it's all the way up to the circle, that would be 100%, like we have 100% accuracy. And you see the different age groups for, various circles. So yellow represents under 10 years old, pink is 11 to 14, the blue is 15 to 21. And you can see that that circle is getting bigger with age. So definitely we're able to classify the motor intent better as the children get older. This is in the unaffected limb. Here is the affected limb. And again, you see that by age, we're definitely seeing an increase in accuracy of our ability to decode the motor intent. The youngest kids are obviously in the center, the oldest kids are in the periphery. So we definitely see a gradient in our ability to classify these motor intents with age. If we go even further, so let's say if we look at the differences between the affected and the unaffected limb. So, let's say we look at our overall ability to decode motor intent in these kids. If we look at the differences between between the two limbs, between the affected and unaffected limb, and we plot that as a function of age. In this case, we're plotting in terms of months. You can see that for the younger kids, like 60 months or so, you can see that there's a greater difference, which is plotted on the y-axis, difference between the affected and the unaffected limb in terms of that accuracy. There's a greater difference for the younger kid that is definitely coming down closer to that zero line with age. So there's definitely less of a difference between the affected and the unaffected limb as they get older in terms of our ability to classify or decode the motor intent in these kids. Okay. So to summarize at least this first part, so far we've tested 46 kids across these different Shriners hospitals. When we talk about our EMG results, the affected average of our decoding or classification accuracy is about 80% in the affected limb, about 92% in the unaffected limb across the 46 kids that we've tested. If we go to ultrasound, it's slightly better. So in the affected, it's 88%, and the unaffected, it is 97%. So definitely some differences between EMG and ultrasound, but for the most part, again, we're able to decode the motor intent in these kids in a hand that has never existed, that they've never actuated quite well using both types of modalities, EMG and ultrasound. And this basically suggests that children born without a hand have a clear motor representation of their missing hand and considerable control over their affected muscles. However, everything that I've talked about so far is really focused on our ability to decode motor intent. We're not talking about really the control that they have over these muscles, really the ability to modulate that control, given visual feedback or given some kind of stimulus or information. So the next part is really talking about the control. Can children proportionally control the position of the residual muscles, basically for a specific type of hand grasp and a hand that they've never actuated, they've never effectively used, and they proportionally control that muscle activation to give us not just relaxation and full activation of those muscles, but somewhere in between. Can they basically modulate that activity? So in this case, we have a fairly simple task. There's a cursor that represents the activation of those residual muscles for, again, a very specific hand grasp, let's say power grasp. And we're asking them to basically move that cursor to different levels. We're asking them to relaxation is all the way at the bottom. So let's say 100%, 0% is relaxation. 100% is full activation, full power grasp. And we're asking them all right, what about a 50% power grasp? Can you basically go somewhere in between? Can you go to 75%? Can you go to 25%? Can you basically modulate that activity for different states of that specific hand grasp? We can get visual feedback and targets to basically direct their performance and examine the precision and accuracy of their control. Can they basically move the cursor to that specific activation amount and basically maintain that for a given period of time? And how accurate can they be? How precise? How long can they basically maintain that activation pattern? And importantly, we can compare between the two limbs, so we can actually see we know that you can do this in your unaffected limb. How does that compare to your affected limb? Are they similar? Are they comparable in terms of their performance? This is a video of the task. So the top plot is a time series of the different target positions that we're giving this individual. So you can see that it starts off fairly high. It's modulating, to different levels, between full activation and full rest. The screen on the over here where you see the black cursor and the blue target, that is what the subject actually sees. So actually see like, trying to move that cursor to different levels based on their activation. In the middle here is the ultrasound transducer on the affected limb. So they're using the activation of their affected muscles to basically move that cursor, again, for a very specific hand grasp. In this case, I believe it's power grasp. And here is the ultrasound imaging, just to give you an idea of like, this is what is actually taking place in the ultrasound image while they're doing this task. So you're going to see all this happening in sync. So you can see the cursor is moving to different levels that we're presenting. They're moving it down now in terms of that, again, very specific power grasp. And they're trying to maintain that level of activation for a given period of time until we move to like the next level of activation. So you can see that this participant is able to modulate that activity quite well. They're definitely able to go between, just rest and full activation, all these different levels in between. And we can have now some fairly good quantification of their behavior, their performance. We can actually, apply some of the things that we know from motor control and motor learning to actually see like, all right, well, how precise are they in terms of the modulation of the activity? We can look at things like reaction time, stability, variance, things like that. And that's what we basically have quantified next, right? So here is the example for one subject. The different colors represent the different levels. So, red represents 17% activation, all the way up to the, let's say, purple represents 83% activation. And he's able to maintain those different levels of activation quite well. This for 4 seconds. The graph on the right, sorry, left, is basically showing the mean activation levels and the standard deviation over the period that we are basically recording over. And you can see that this particular subject is able to maintain those 5 different levels of activation quite well, and they are all statistically different from each other. So they're able to maintain from 17% to 83% activation quite well in terms of this very specific hand grasp pattern. That's the results for one subject. Here is a different subject. You can see they had a little bit more difficulty in the top, the highest activation level of 83%. But the other 4, they're able to maintain statistically different activation levels just fine. So 4 out of the 5 for this person. And for the next participant, similar to the first, they're able to do 5 different levels, all statistically different from each other. So again, this gives you a sense that they're able to actuate these muscles, they're able to modify the activation in these muscles quite well, and to statistically different levels of activation. Again, this is for a very specific hand grasp, so it's not random activation, and it's also, again, in the hand that's affected, so they are missing a hand since birth. Yes.
So just for clarification, when you're saying activation, is this a percentage of like MVC, or is it just a percentage of the completed action? Like 100% is fully power grasp closed, 0% is full.
That's the question. This is a percentage of the completed action.
Okay.
Yeah.
Thank you.
Thank you for asking. All right. So for all children, the mean stable cursor position was significantly different for at least 4 out of the 5 proportional levels tested. And that was, across the 46 subjects that we've tested so far. If we look at a couple things in terms of the dynamic properties of their ability to control, these muscles, we can kind of go to the motor control literature to look at some similarities. So in typical arm reaching movements, if you have someone to come to the lab and basically make point-to-point reaching movements, they are close to a minimum jerk trajectory. And basically all that means is that they minimize the acceleration changes over the duration of that movement. Jerk, I'm sure almost everyone probably in the room knows, but jerk is a derivative of acceleration. And if you look at these point-to-point reaching movements, if you're moving, from one position to some peripheral position, you have some systematic change in your position, systematic change in your velocity, systematic change in your acceleration. And this framework is useful because it tells you, or if you want to do smooth, accurate movements, this is one way that you can do it, in one way that's been shown that you can do it basically in the lab. And we can see, like, as subjects are basically moving this cursor from different positions, different levels, does it follow something similar to what we see in normal human movement, something we see somewhat similar in these point-to-point reaching movements. So we can compute the ideal movement trajectory and the velocity profile using this kind of framework and see how well does that match, or how well does that kind of simulate what we see in these kids as they're basically trying to control the residual muscles in their limb. Does it follow somewhat similar properties? So going back to what I said before, if we have people come in, able-bodied subjects, if they make point-to-point reaching movements, let's say we sit them down in front of a digitizing tablet, we have them, basically grab a handle, move a cursor from one position to another. We can look at things like their velocity profiles. And what you see is a systematic change in the peak velocity. So the blue represents a short distance, the purple represents a longer distance. You can see that the peak velocity of those movements is definitely increasing with distance, which is what you would expect from a minimum direct trajectory. Also, the duration of those movements is also increasing. So the longer the distance, the longer the duration of the movements. That's, again, similar to what you would see for minimum direct trajectory. We did this study in able-bodied subjects using ultrasound, let's see, 2 years ago. So it's the same kind of task where they're basically asked to move the cursor between these different levels for a very specific hand grasp pattern, in this case, again, power grasp. And when we look at the movement trajectory, so when they're moving the cursor from different levels in this task, you see somewhat similar aspects of minimum direct trajectory. You definitely see an increase in the peak velocity. You definitely see an increase in the duration for different longer distances that the cursor has to travel. So there's definitely some similarities using ultrasound imaging that we see in normal reaching movement behavior. If you plot these things out, so if you look at all the different subjects, if you look at, let's say, the normalized time, so the time it takes to actually reach a target, and you plot that as a function of the peak velocity, you definitely see the systematic relationship for different distances. So the blue, again, is the shortest distance, purple is the longest distance. You definitely see this nice relationship between those two things, the time and the peak velocity. If you look at the cursor task with ultrasound, messy, but similar relationships. You definitely see a increase in the peak velocity that coincides with the increase in the time to the target. So somewhat similar to what we see in normal reaching movements. If we go back and look at our kids, if we look at the pediatric population, again, in this task where they're trying to move that cursor to different targets, different activation levels, we can look at things like the velocity profiles. So red here represents the shortest distance, purple represents the longest distance. You can see that the peak velocity absolutely scales with movement distance, which is what you would expect. This is in the unaffected limb. If we look at the affected limb, you see very similar relationships, again, between the shortest and the longest distance, and again, between the affected and the unaffected limb. I said before, for different longer distances, you should see an increase in the peak velocity. So that peak velocity should scale with distance. If we look at our kids, you absolutely see that as well. So red is the shortest distance, purple is the longest. You definitely see an increase in peak velocity of that cursor movement for the unaffected limb, similar to what we see in the affected limb. If we go even further and compare between the two limbs, so within a child, we can plot the affected and the unaffected limb against each other in terms of these different parameters of the task. If we look at the affected peak velocity on the y-axis, the unaffected peak velocity on the x-axis, you can see again that there's really no difference between the two limbs because they both are hugging this unity line, the black dashed line down the middle there. And again, it's for different distances. So red shortest, the purple is the longest. So really no difference in the peak velocity between the two limbs. Again, this is for a hand since birth that hasn't existed. There was no significant difference between the affected and the unaffected limb in this case. We can also look at things like the distance. So when they maintain this this position level of the cursor, we can look at the affected limb versus the unaffected cursor position within each child. Blue here represents the shortest distance, the green represents the longest distance, and again, we see no difference between the affected and unaffected limb. They're both basically riding that unity line, showing that there's very little difference between the two limbs. So overall, we're not seeing really any significant differences between the two limbs in this control task, this motor control task, for again a very specific hand grasp pattern, different activation levels. Right. If I were to conclude, right, for this, so children with UCBED, children with this unilateral congenital limb difference have robust control over the muscles of the residuum. We are able to use ultrasound and EMG to basically show that they can act at least 10 different hand grasp patterns with high specificity and reliability. I should have also pointed out the training that these kids have before we test the ability to code or the ability to do this modulation task is minimal. We're talking about that day they're able to do these things. So it's not like they've been trained for weeks or months. It's really within tens of minutes, 30 minutes. Children with UCBED can use sensory feedback, and that task that I showed with the different levels, different targets, sensory feedback to modulate the control, the state of their affected muscles. The untrained motor abilities of the affected limb do differ from the unaffected limb, but they also show many similarities. So when we talk about the structure of the muscles in the two limbs, that's definitely different. But in terms of their ability to control those muscles, that is fairly similar. Key features, like when we talk about what we would expect from minimum trajectory, those kind of characteristics of typical motor behavior definitely exists in the control of the affected limb, and we're definitely looking more into that in terms of how we can utilize that for controlling prosthetic devices more effectively. If I were to briefly just talk about some of the future things that we are working on, or some of the future directions. So the first thing is we need to finish the study. Like I said, we're up to 46 out of 50 kids. The goal is to get 4 more in the next couple months. And we will have a fairly complete data set, I think, by the time we get to like midsummer. We're going to examine things like age, sex, limb length, prosthesis use in terms of their control, their ability to decode different types of motor intents, and also their ability to control the modulation of that activity in their affected and unaffected limb. Ultimately, we're trying to build better devices. So, if we have better control signals, we should be able to control these devices in a more effective way. So we're trying to implement that in the real-time control of physical prosthetic, hands. So instead of moving cursors on screens, actually controlling physical devices. This is one of the graduate students, Marcus, who's put in a tremendous amount of work into this project, and he's working with one of the participants. And you can see that this is with EMG. So the EMG sensors are on the affected limb, and they're basically using the EMG signals to control one of our experimentally developed hands. And they're just basically going through different types of hand grasp patterns. You can see that he's doing a fairly good job. I think there's one where he has to correct it, right? So he went to pinch, but he was trying to do a key grasp. So able to basically go between these different hand grasp patterns using EMG. We're also using ultrasound imaging to do the same kind of studies. So we have 2 hands in this case, right? This is for the unaffected limb, and he's going through the different motions and the unaffected limb, and basically controlling that robotic hand. We also are doing the same kinds of things with the unaffected limb to see how they compare in terms of this real-time control of a physical device. So using EMG, using ultrasound, we're looking at real-time control of these devices. And of course, the ultimate idea is to expand this proportional control in multiple motions simultaneously. And that is a challenge, but something that we are actively looking into to basically try to do. 2 more slides. One of the questions I usually get is about like what happens if you actually have someone who has two limbs that are congenitally missing. So we are doing imaging studies. We're not able to answer that question exactly, but basically, what does that representation look like when it comes to the brain? There has been some work using fMRI out of Tamar Makin's lab that has suggested, if you look at the representations of the digits in folks that have a traumatic or congenital limb difference. So if you look at the representation of the 5 digits and the sensory motor cortex, if you represent this in some two-dimensional kind of space, you can see that there's high separability if you're an able-bodied subject. So that the 5 digits are actually represented in very distinct ways in the sensory motor cortex. However, if you go to someone who's had a traumatic limb difference, so if they had a hand and unfortunately lost it at some point, you can see that the separability, the ability to basically disassociate the 5 digits is a little bit less so, a little bit more clustered. And if you go to someone who has a congenital limb difference, you see that the organization, or at least the structure of that, is heavily implied that doesn't really exist nearly as well in a congenital limb difference subject. These are all adults, I should say. And again, they're looking at specific representations of the digits. One of the things that we recently were awarded from Shriners Hospital is to do an fMRI study looking at the kids with congenital limb difference. And instead of looking at the individual digits, actually look at the grasp patterns. So we're going to have the kids actually do these specific grasp patterns in the scanner to see what the representation of those grasp patterns are in the sensory motor cortex. So we're really excited about that. As you can imagine, doing fMRI studies with kids, it's going to be a challenge, but it's something I think that'll be really interesting, give it some insight in like how the actual neural motor control, how that relates to what we see in the periphery in terms of the control over those muscles. The last future thing is we're not totally against, actually studying adults. We've had some adult studies too. In this case, we're looking at folks that have targeted muscle innervation. So these are adults that have had some kind of traumatic event. They've unfortunately lost a limb, and instead of basically having the nerve endings basically grow and create somewhat painful neuromas, we have surgeons who are, one of many across the United States that are doing surgery. I apologize. I should have warned people for that. I'm sorry that that's bloody. But in terms of preventing really painful neuromas, what they do is a surgery called targeted muscle reinnervation, where they basically take those nerves and they reinnervate them into different muscles. So now you have nerves that control the hand that are now controlling parts of the limb, sometimes even the chest muscles. And you have very unique muscle activation patterns in these individuals. So when you ask them to actually do certain types of hand motions, there are very unique patterns of muscle activations, and they're recovering residuum of their limb. So we can look at what those muscle activation patterns look like, and can we use those for prosthetic control? Because typically these kind of surgeries, or at least the vast majority of these surgeries, are really done for sensory or at least pain prevention, and not really for controlling prosthetics. But we think that we can use those muscle activation patterns, and we can study them really well to have motor control signals. In addition to the actual form, sometimes these are innervated into the chest muscles, and we've had some interesting studies where we looked at ultrasound imaging of the chest to actually see how those chest muscles are actually decoding different types of hand motions, like grasp, power grasp, pinching, things like that. Not just with ultrasound, but also with EMG. We're seeing somewhat similar results in terms of the control over those chest muscles and how they relate to hand grasp patterns. To just get some quick acknowledgements, I just want to definitely thank my collaborators, John Schofield in the Mechanical and Aerospace Engineering Department. I couldn't do this work without him. He's been a fantastic collaborator and a great friend. Dr. Anita Bagley and Dr. James both work at Shriners Hospital. Again, we couldn't do that without their help and all their clinical insights. Marie is the clinical coordinator at Shriners. Justin, Marcus, Eden, and Josh are the graduate students who, of course, have spent a tremendous amount of time and energy traveling to these different sites, testing these kids, and everything I presented is really their work. I should definitely thank our funding sources like the NSF, the NIH, and also Shriners Hospital, which has been a big driver and a big supporter of the work that we've been doing. Again, I want to point out Justin and Marcus because this was their thesis work. Most of what I presented is their thesis work for their graduation. Members of our research team at the various sites, so Portland, Chicago, Greenville, we wouldn't be able to do it without the cooperation of all these different hospitals and the folks that basically work there. And I should also, be remiss if I didn't thank the kids for putting up with our experiments and their families for entrusting us with, basically working with them and testing them and getting a really good handle on their abilities and, hopefully trying to serve their needs going forward. And with that, I will stop and take any questions you may have. Lena.
Thank you. There's so much motion in their video that's really impressive. I want to know what the neural mechanisms underlying their ability to retain ability to control these muscles is, and
You and me both.
do you really need to have a representation of individual fingers, or do you think it's a higher up representation of a task level type of?
I think it's a high representation. I don't think it's individual fingers. I think that, and this is not to throw shots. I think Tamar Bacon would probably argue against that, based on what they've shown before. But I do think with the kids, we have a unique opportunity to answer that question at least more definitively. So I suspect that it's more of a higher level as opposed to individual representation of those digits. But my hope is in 2 years I can tell you.
Is that hardwired or?
Oh, I don't know. The question before about if there was what would this look like for individuals that had both limbs, a congenital difference? I find that question fascinating, and I asked Shriners because they have the whole network of all their records, and I think there are just a couple kids that exist, and I've pleaded that they let us do the same kind of study because that question comes up again and again, and you know, we talk about like, is it transfer from one hemisphere to the other? Is it some kind of experience like dependency? Like, I wish I had the answer, but I'm trying. Yes.
Hi, I'm curious about the let's accuracy for the younger kids, and even for the unaffected ones.
Yeah.
Is it due to smaller size and smaller motion? And if the ultrasound image, is it the resolution is not good enough, or variability of the what are the major
Right. So perhaps it's not so much like their ability to do this is worse, but maybe like there's actual like technical or at least like more analysis or quantification aspects that are like the problem, and that's why we're getting lower accuracy for those kind of kids. Is that what you're implying, or?
But what aspects? Is it the larger stuff? I mean, you know, the probe is too large for them, or the resolution of the image, or what technical issue?
You know, there's one thing I didn't say is like, you know, the youngest that we're testing, and this is something that the clinicians basically said was like, you can't go below 5 because they're not going to be able to pay attention long enough, or at least like give you, you know, enough of their attention for long enough periods of time to do this. Some of the studies that I presented, like some of the data sets are like 3/4 of the data set because they basically petered out after like, you know, time. So there are some caveats that I definitely should give when it comes to like, you know, age dependency or kids, you know, these results for kids. The things that you're suggesting about like, you know, the size, I think there was a question about, you know, what's actually happening to the muscles and things like that. Those are, I think, excellent questions. And there are a number of things I think that we need to, you know, from a basic science point of view, sort out and study tremendously, and like more of a technical, these technical aspects of like, you know, what does this mean? What are these different aspects? How does that actually affect our ability to decode and things like that? So I don't have a definitive answer for you, but I know that those are important aspects that we need to look into. You're right. Yeah, I think I could say this. The thing that they want us to do is build a device. They want us to build a prosthetic. Everything that you're saying are all the technical things you need to figure out first before you put ultrasound sensors in a socket and then put it on someone's like residual limb. So I think there's still a ways to go. I think these are promising results, that these definitely seem to be effective and working, but all the technical aspects of it are, you know, we're still a number of years away until we actually are going to be able to do that. Back for, yeah.
The first one is, first of all, like really interesting thought. Good work. The first question is, how does like one hand generalize to the other? So for example, you know, like if you have a decoder that you train for the unaffected limb, if you just apply the same decoder without any training on the data or the readings that you get from the affected one, how good is it in generalization? And you can even take this question further. How does it generalize, you know, like across
the kids,
people, you know, like on the same age or even at different ages?
Yes.
And it's linked to the representation question that I was asking earlier, because can we have some form of like a library of or like even a lower level representations that are working with multiple people and, you know, like plug and chug basically.
So those are excellent questions. The first thing I would say is like with a data set of about 50 kids, those are things that we can actually answer, or at least like, you know, go about like figuring out how best this generalizes, or how what are the characteristics that are universal as opposed to like fairly individual. So yes, so that's an excellent question. The other thing you asked this before, one of the things that we actually also did is do RSA analysis to actually look at this in like some like 3-dimensional space. So these represent like, you know, the different handgrass patterns and how they actually move within a, you know, if you're doing a power grass, how does that actually change over time within this 3-dimensional space? So when you're asking about like, you know, how separable they are, or at least how, you know, distinct they are, you can see that they definitely cluster in different areas in this 3-dimensional space, and definitely have fairly specific. Is it gonna do it again? No, it's not. Fairly specific trajectories. Oh, it's not gonna do it. It's not gonna do it. All right. Fairly specific trajectories that they travel. So
Might be.
it might be the best. I guess maybe the way that I can answer that question is like, you know, there's all these basic science questions. This goes back to the question before. There's all these like engineering, you know, quantification, you know, anything you want to throw at this, like information theory, you know, various aspects that I think are relevant and very interesting in terms of what and it's not to say anything negative about Shriners, in terms of what Shriners is interested in, right? They want to make sure that everything that we're doing is, in the direct, and rightfully so, everything we're doing is directed towards meeting the needs of these kids. And they let us do engineering occasionally. So yeah, everything that you said is absolutely things that we need to do. Yeah. Yes. Sorry. Yep.
Necessarily, like it's not necessarily their non-dominant and just because they've lost it, how could that affect them?
So that's an excellent question too. I don't have a video of it. I wish I did. So one of the things that we did is first we did it with my kids. So I should say that my kids were the guinea pigs first. So we put GoPro cameras on their forehead, and we had them basically interact for like hours with like the GoPro cameras to basically see like how do they actually like use their limbs, what are the most important grasp patterns, like what do they actually how to actually use their hands on a day-to-day basis. We did that. We published a short paper on that. The next thing was to send some of these kids home with Ukbed with those GoPro cameras on their head. They're still going through all the data and all the analysis. But to your question is like, how much do they actually use their unaffected limb? How do they use it? Is it something that they in terms of like their day-to-day interactions, is it something that's more of a support or more of like an active participant in like these kind of interactions? Those are things that we should have the information to answer. And I think it gets at your question of like dominant versus non-dominant, and like especially if we compare it to able-bodied kids, like how do these things compare? So we definitely have the data. It's just a matter of actually getting through it to answer those kind of questions. But that's an excellent question too.
See how it's something that's important, right? So I could see if you're deficient limb or you're dominant hand, you would use that limb differently than if it were your non-dominant.
I can see that too. But of course, these kids are also born with that limb difference. So it's not like something traumatic. Yeah. Yeah. It's an excellent question. I assume that's probably not the same. That would be my guess. But we definitely have the data to basically answer that question.
That's why you said one question of it.
Are we at time? I'm sorry. Okay. Oh, it is 12. Okay, yeah, right. Yeah.
The high level question again, and I feel like you might have all the pieces, or most of the pieces to answer it. But the question is something like, can you say how much of a neural control adapts to physiology versus vice versa? Because you're studying children, and they're developing, and you have a machine learning algorithm that measures tries to predict movement based on EMG and ultrasound. So do you really have enough pieces to try and answer like?
Maybe I'm making this the question I want to answer. But are you talking about like bimanual communication between like the algorithms we use and the actual kid, or are you talking something different?
In I guess humans would say like, how our physiology is continuously changing, like our bodies are changing, and the neural control is changing.
Yes.
So like which one is adapting to which?
I see.
Because you're studying children who are developing and you're trying to predict behavior using these algorithms. I'm wondering if you have enough information.
Okay. I think there are two things that we want to do, but we haven't done exactly yet. So like most of the motor learning kind of studies that you could think of, like retention, generalization. If you have some of these kids we've seen like maybe twice, maybe 3 times over the course of a year, do things get if you study like a single child over that time period, do those things within that child get better? Are they get more generalizable, or do they retain some of that information better? So those are kind of things that I want to do, that we want to do. The second thing is the fMRI study that I talked about. When you talk about the neuromotor control, you're absolutely right. The developmental grant is only 2 years. It's a fairly small study. We can only do about probably like 12 to 15 kids. We'll have some information, but we won't have a time series of that information. So we won't be able to like scan kids over multiple years, and we won't be able to definitively answer what you're getting at. But that's exactly what I want to do. right? Like if you actually had a 5-year period, you could actually test somebody who's like 5 years old, 10 years old, and actually show within that subject like what's actually happening at the neural sensory motor level. Like that would be fascinating. Hopefully we'll be able to do that. Yes. Cool. Good. Okay.